Good morning, students. In today's lecture, we will explore the classification and morphology of bacteria. Understanding bacterial shapes and structures is essential in microbiology because these characteristics help scientists identify different types of microorganisms. Before studying classification, let us first understand what bacteria are. Bacteria are prokaryotic organisms, which means they do not have a true nucleus. They are single-celled microorganisms that are extremely small and can only be observed under a microscope. Another important feature is their distribution. Bacteria exist almost everywhere on Earth, in soil, water, air, food, and even inside living organisms. Bacteria play an important role in food and dairy systems. On the beneficial side, some bacteria help produce yogurt and cheese. They are responsible for fermentation, which converts lactose into lactic acid. This process also contributes to flavor development in many dairy products. However, not all bacteria are beneficial. Some bacteria cause food spoilage, foodborne illness, and safety concerns. Therefore, understanding bacterial behavior is very important in food processing. Let us now examine the structure of a bacterial cell. A bacterial cell contains several essential components. First, the cell wall, which is mainly made of peptidoglycan. This structure provides shape and protection. Second, the cell membrane, which controls transport of nutrients and energy flow. Third, flagella, which help bacteria move. Finally, some bacteria possess a capsule which provides protection and helps the organism survive harsh conditions. Bacteria are classified using several criteria. The first criterion is shape. Bacteria can appear as cocci, bacilli, or spiral forms. The second criterion is arrangement, such as pairs, chains, or clusters. The third criterion is gram staining, which divides bacteria into gram-positive and gram-negative groups. The fourth criterion is metabolism, which indicates whether bacteria are aerobic or anaerobic. These characteristics help microbiologists identify bacterial species. Let us begin with cocci, which are spherical bacteria. The arrangement of cocci depends on how the cells divide. Diplococci appear in pairs. Streptococci form chains as cells remain attached after division. Staphylococci form clusters that resemble bunches of grapes. These arrangements are determined by the planes in which bacterial cells divide. Next, we look at bacilli, which are rod-shaped bacteria. Bacilli can occur in several arrangements. A single bacillus exists as an individual rod-shaped cell. Diplobacilli appear in pairs. Streptobacilli form chains. Coccobacilli are short rods that appear almost spherical. Common examples of rod-shaped bacteria include Escherichia coli and Salmonella. Another group of bacteria includes spiral forms. There are three main spiral shapes. Vibrio bacteria are comma-shaped, such as Vibrio cholerae. Spirillum bacteria have rigid corkscrew-like structures. Spirochetes have flexible, tightly coiled structures. These different shapes are useful for identifying microorganisms. Bacterial arrangement patterns serve as diagnostic clues. The arrangement of cells is determined by the pattern of cell division. For example, cocci may appear in pairs, chains, clusters, or tetrads. Rod-shaped bacteria may occur singly, in pairs, or in chains. Therefore, cell arrangement acts like a fingerprint for identifying bacterial species. Another important classification method is gram staining. Gram-positive bacteria have a thick peptidoglycan layer in their cell wall. They do not possess an outer membrane and appear purple after gram staining. Gram-negative bacteria have a thinner peptidoglycan layer but possess an outer membrane. These bacteria appear pink after staining. This difference in cell wall structure is important in microbiology and medicine. This slide provides a visual summary of bacterial classification. Bacteria can be identified based on three main characteristics. First, their shape, such as cocci, bacilli, or spiral forms. Second, their arrangement, including diplococci, streptococci, staphylococci, and streptobacilli. Third, their staining characteristics, which distinguish gram-positive from gram-negative bacteria. Together, these features help microbiologists accurately classify microorganisms. To conclude, bacteria are microscopic, single-celled organisms with diverse shapes and structures. Their classification depends on shape, arrangement, gram staining, and metabolic characteristics. Understanding bacterial morphology is essential for studying microbiology, food safety, and dairy technology.